Hey there! Ready to jump into some cutting-edge medicine. Today, we're going deep into oligonucleotide therapies. Those DNA or RNA strands that can zero in on specific genes.
Yeah, really fascinating stuff.
What's really wild is how one tiny chemical change can completely change how these therapies work. We're talking making them more stable, last longer in the body, and work even better.
It's true, those tiny tweaks at the molecular level can make a huge difference.
And for this deep dive, we've got this article from Assay Genie. They're all about biotech tools, so we know it's legit.
Oh yeah, for sure, they're a great source.
Okay, so we keep saying small chemical change, but what is it exactly?
Well, it's called a phosphorothioate modification.
Phosphorothioate.
Yeah. Basically, you've got the backbone of DNA, or RNA. It's like a chain of phosphate and sugar molecules. Now, imagine just swapping out one oxygen atom for a sulfur atom in that phosphate group.
So one tiny swap, but what does it actually do?
It's like giving those therapeutic molecules a shield, protecting them from the body's natural defenses. Our bodies are really good at breaking down foreign molecules, including these helpful therapies.
Right, they don't stick around forever.
Exactly. But that sulfur atom, it makes them much harder to break down.
Oh, so it's like they can actually get to where they need to go and do their job.
Exactly. And that's where the real magic happens. It's like, by making them tougher, we see three main benefits that really change how these therapies work.
3. All right, lay 'em on me. What makes the sulfur swap so special?
Okay, number one, increased nucleus resistance.
Nucleus resistance.
Yeah, so our bodies are packed with these enzymes called nucleases that break down DNA and RNA. It's like they're constantly on patrol, cleaning up anything that looks out of place.
So our bodies are working against these helpful therapies.
In a way, yeah. But with phosphorothioate, the therapies become much tougher for nucleases to digest.
So it's like they can resist the cleanup crew.
Exactly. And that stability means the therapy can stay in the body longer, increasing its chances of hitting the target and doing what it's supposed to do.
That makes a lot of sense. But is there more to it than just lasting longer?
Of course. Number 2 is something called enhanced binding affinity.
Okay, that sounds complicated.
It's not really.
Yeah.
Just think of it like this. The phosphorothioate modification helps these therapies grab onto their targets more effectively.
So they're stickier.
Sort of, but it's more than that.
What kind of targets are we even talking about?
We're talking about specific mRNA sequences.
mRNA.
The messengers that carry instructions from our DNA to build proteins. These therapies can target specific mRNA to block their effects or change how they work.
Wow, that's crazy precise. Increased stability, enhanced binding. What's number 3?
Number 3 is improved cellular uptake. Basically, phosphorothioate makes it easier for these therapies to get inside cells.
Like a VIP pass.
Kinda. It helps them slip in and get to work.
So all these benefits, the stability, the binding, the uptake, they all work together.
Exactly. And that's how we get much more effective therapies.
So this isn't just a minor tweak, it's like changing the whole game, right?
You got it. But how exactly do these molecules actually work their magic?
Yeah, let's get into the nitty-gritty. How do they actually work at the, molecular level?
Well, there are 3 main ways these phosphorothioate molecules are being used. Each one has its own unique mechanism. The first is called antisense oligonucleotides, or ASOs.
ASOs.
Yeah, imagine them like special agents sent in to take down a specific target.
Ooh, that sounds intense. What are they going after?
They target specific mRNA sequences, those messengers that carry instructions from DNA to build proteins. But here's the thing. When an ASO binds to its target mRNA, it basically destroys it.
Oh, so it's like intercepting a bad message before it can even cause trouble.
Exactly. And because these ASOs have that phosphorothioate modification, they're way more stable and effective at doing their job.
So they're like super agents.
You could say that. This makes them super helpful for treating diseases caused by, too much of a certain protein.
Wow, so it's like shutting down a bad gene before it can cause any harm. What about the second mechanism?
That one's called RNA interference, or RNAi.
RNAi.
Yeah, and it's like silencing a noisy neighbor.
Okay, how does that work? I'm picturing earplugs or something.
It actually uses these tiny molecules called small interfering RNAs, or siRNAs.
siRNAs.
Right, and they're also modified with phosphorothioate.
Of course, gotta have that stability.
Exactly. So these siRNAs, they target specific genes and basically shut them down completely.
So instead of destroying the messenger RNA like the ASOs, they silence the gene itself.
You got it.
Wow, that's a completely different approach. But I guess the goal is the same. Control gene expression.
Precisely. Both ASOs and RNAi are super powerful tools, and which one is used depends on what the goal is for treatment.
It's amazing how scientists are figuring out how to actually control our genes like that. So what's that third mechanism you mentioned?
That one involves aptamers. They're like little molecular detectives. Yeah,
Detectives.
they're designed to bind to super specific molecules with amazing precision. And, you guessed it, phosphorothioate helps them stay on the case longer.
They sound like real pros.
They are. What's cool is that they can be designed to bind to all kinds of targets, from proteins to small molecules.
So they're versatile.
Exactly. Makes them valuable for therapies and diagnostics.
Wow, that's pretty incredible. Okay, so we have these three mechanisms, all using phosphorothioate to be more effective. It sounds like this stuff is already changing medicine as we speak. Are there, like, any specific examples of how it's being used right now?
Absolutely. Let's start with genetic disorders. One area where these modified oligonucleotides are really making a difference is in treating spinal muscular atrophy, or SMA.
SMA, yeah, I've heard of that.
It's a really tough genetic disorder that affects muscle development.
Yeah, it's especially heartbreaking because it often affects kids. How are these new therapies helping those families?
Well, SMA is caused by a problem with a gene called SMN1.
SMN1.
Right. This gene is responsible for making a protein that's essential for motor neurons, the ones that control muscle movement.
So without that protein, the muscles can't work properly.
Yeah, the motor neurons die.
Yeah.
And that leads to muscle weakness and wasting.
That sounds devastating.
It is. But there's another gene called SMN2 that can kind of step in and help.
So it's like a backup.
Kind of, but the problem is that SMN2 makes a slightly different version of the protein, and most of it doesn't actually work.
Oh, so close but no cigar.
Yeah, and that's where those phosphorothioate ASOs come in.
They can fix a backup.
In a way, yeah. They can target the SMN2 gene and modify its RNA splicing. Basically, they trick it into making more of the functional protein.
That's incredible. It's like fine-tuning the backup generator to actually work. Are there any other genetic disorders where this approach is showing promise?
Absolutely. Duchenne muscular dystrophy, or DMD, is another one.
DMD.
It's another serious muscle wasting disorder.
Sounds similar to SMA.
It is, in a way.
Mhm.
DMD is caused by mutations in a gene called dystrophin.
Dystrophin.
Yeah, this gene is super important for muscle fiber strength and stability.
So it's not working right.
Exactly. The muscle fibers get damaged easily, and that leads to muscle weakness.
So how are these modified oligonucleotides helping with DMD?
Okay, so with DMD, scientists have actually developed ASOs that can skip over the mutated parts of the dystrophin gene.
Skip over them.
Yeah, so the cells can make shorter version of the protein, but it still works.
So they're basically editing the instructions.
It's called exon skipping.
Exon skipping.
Yeah, and it's showing really promising results in clinical trials.
That's awesome. So these therapies can target specific genetic problems and actually change their effects. It's like having a molecular toolkit to fix these diseases. Are there other areas besides genetic disorders where this is making a difference?
Oh, definitely. Another exciting area is oncology, the fight against cancer.
Cancer. That's a tough one. I can see how this technology could be a game changer there.
Absolutely.
So how are they using this stuff to fight cancer?
Well, one way is to use ASOs to, directly target the cancer cells.
Makes sense.
For example, some ASOs are designed to block the production of proteins that cancer cells need to grow and survive.
Oh, so like cutting off their food supply.
Exactly, like taking out a key factory that the cancer relies on.
Smart.
And then another approach is to use ASOs to actually boost the body's own immune response.
Oh, so our bodies are already fighting, but these therapies can help them fight even harder.
Right. There are ASOs being developed that can block the signals cancer cells use to hide from the immune system.
So it's like making the cancer visible so our immune system can see it and attack.
Exactly. It's like removing their camouflage.
That's pretty cool.
Yeah. And beyond that, phosphorothioate can also make other cancer treatments work better.
Really?
Like, for example, they can be used to make sure chemotherapy drugs go straight to the tumor cells.
Oh, that's awesome. Less side effects and more effective treatment. So, wow, so many ways to use this in the fight against cancer. It really seems like this technology is opening up a whole new world for treating all sorts of diseases. But it's not just genetic disorders and cancer, right? You mentioned cardiovascular disease, too.
Right, right. One really promising area is using ASOs to target a protein called PCSK9.
PCSK9.
It plays a role in regulating cholesterol.
Cholesterol, that's important for sure.
Yeah. By blocking PCSK9, we can increase those good cholesterol receptors in the liver.
So basically, it's helping the liver do its job better.
Precisely. And, get this approach has already shown some amazing results in clinical trials. People's bad cholesterol levels drop significantly.
That's huge. Could really help reduce the risk of heart attacks and strokes.
Absolutely, it's a big step forward for sure.
It's really incredible how much impact this one modification is having. But, with all this amazing potential, are there any downsides? Like, what are the challenges?
Well, yeah, of course. Like with anything new, there are things to watch out for. One thing is, making sure they target the right thing.
Right, because if it messes with other stuff, it could cause problems.
Exactly. It's called off-target effects.
Off-target effects, got it.
Yeah, so like, they're designed to be super specific, but there's always that risk.
So rigorous testing is super important.
Right. Oh, absolutely. To make sure it's safe and it actually works. Another challenge is the cost. Developing and making these therapies can be really expensive.
Yeah, that's a big one. It needs to be accessible for everyone. But even with those challenges, it sounds like the future for this technology is super bright. What's next? What are scientists working on now?
Well, there's always something new. Researchers are constantly looking for ways to, make these therapies even better.
Better? How?
Well, for example, they're working on combo therapies, like mixing phosphorothioate with other modifications. Imagine being able to really fine-tune how these therapies work.
That's wild. What else is in the works?
There's a lot of talk about using phosphorothioate to deliver gene editing tools.
Gene editing, like CRISPR.
Exactly.
Wow, that's next level stuff.
Yeah, it's pretty amazing.
So using this to make gene editing even better, that's huge.
It could be revolutionary.
It's mind-blowing to think that one tiny tweak, swapping an oxygen for a sulfur, is having such a huge impact.
It really is. It just goes to show how powerful chemistry can be.
And the amazing ingenuity of the scientists working on this.
Absolutely. It's a great example of how research and innovation can really make a difference.
So this is just the beginning then.
Oh, yeah, for sure.
That's super exciting. Well, listeners, we hope this deep dive has sparked your curiosity. The world of science is constantly moving forward, and we're here to break it down for you. So until next time, keep exploring and stay curious.